In this video, we will discuss this M1 macrophages and pancreatic cancer cells interactions in detail. In the previous video, we have understood that how the spatial transcriptomics and scRNA reveals the cellular interactions surrounding the pancreatic tumor cells with the pancreatic tumor cells, and we have find out that epithelial cells with the M1 macrophages has the highest interactions compared to the other cells that are surrounding the pancreatic tumor cells. So in this video we will understand what are the changes that occurs within the pancreatic tumor within the macrophages that cause its high interactions with the pancreatic tumor cells. So first, in order to validate their results from the previous experiment, what they did is they did the immune immune infiltration analysis of the pancreatic tumor cells that are publicly available, and in this they have find out that the macrophages infiltrations in the pancreatic tumor cells is lower compared to the case of this healthy pancreatic cells. Once validating their hypothesis that they have observed from the previous experiment that macrophages infiltrations are lower, they wanted to check what are the changes that occurs within the macrophages that cause its behavior to act like this. So the initial step is that they took the differentially regulated genes in the macrophages of the pancreatic tumor cells compared to the healthy pancreas, and then do the bioinformatics analysis in a way that they have filtered out the genes, and finally they come up with the 8 candidates. Out of the 8 candidates, they shown here the 4 candidates, and in order to understand how they filtered out the differentially regulated genes to come up with these 4 genes, you can refer to the supplemental figures, and in order to go to the supplementary figures, you can use the link that has been provided in the description. So once they identified that these 4 genes could be responsible for this macrophages behaviors, then they check the p-values, and for p-values for this MMP14 and MIF is higher than 0.05, so they reject these p-values, and then thereafter they left with 2 candidates, RPS18 and IRF7, and they check that IRF7 is not hazardous whereas RPS18 is hazardous. So their final candidates was this RPS18. So thereafter they move to check. Thereafter they move to check this RPS18 genes regulations within the macrophages, and since the RPS gene is responsible for this IRF7, they check its level as they move further in the experiment, and thereafter they did the bioinformatics analysis where they find out that as they move close toward the pancreatic tumor cells, or as the pancreatic tumor cells gets more purer, they see the reduced level of IRF7, so indicating that RPS18 upregulations might be causing this IRF7's down regulations. And also when they check with the macrophages, in the macrophages also they see that macrophages infiltration levels increases as its macrophage infiltration level increases. With the increase in the infiltration level, they see the macrophages amounts are also increased. So this indicates that cells interactions with the M1 type macrophages could be occurring, because as the tumor microenvironment cells approaches close to the tumors, the amount of macrophages in the tumor microenvironment increases. That is shown in this figure. And after doing this bioinformatics, they wanted to check whether their results are true, or they wanted to check their hypothesis. So what they did is they took the pancreatic tumor cells and also the tumor cells in the tumor microenvironment, that is normal cells are surrounding the pancreatic tumor cells, and check the level of this IRF7, and they see that in the M1 macrophages the IRF7 levels are higher in the tumor microenvironment case, or the normal cells surrounding the pancreatic tumor cells compared to the pancreatic tumor cells, indicating that IRF7's modulations might be occurring as they move or interacts with the pancreatic tumor cells, or as they infiltrate towards the pancreatic tumor cells. However, for the M2 macrophages, these IRF7 levels are unchanged, and for similar for the epithelial case. And After validating that it's the IRF7s that is modulating within the M1 macrophages, they again did similar experiments with normal cells and the pancreatic tumor cells, and in this case they have find out that in the normal cells or in the normal patients that does not have the pancreatic tumors, they see the high level of IRF7, whereas in the case of this pancreatic tumor cells the IRF7 levels are down, whereas for this M2 macrophages and epithelial cells there is no change occurs, so indicating that it's the IRF7's modulations that might be causing its high interactions with the pancreatic tumor cells, and that might lead to the cellular changing in the pancreatic tumor cells. So further validate their results, they isolate the CD163 positive cells and CD86 positive cells, which is the markers for the M1 macrophages and M2 macrophages respectively. I am 90% sure, however, you need to validate these markers either via internet or go through the reference link. It could be reverse also, but I'm 90% sure the CD163 is the marker for M1 macrophages and CD86 for the M2 macrophages. So once they identify, separate out the cells for this M1 macrophage and M2 macrophage, they check the IRF7 using the Western blot, and they can see that cells around in the tumor microenvironment of the pancreatic tumor cells has the high IRF7s compared to the case of this M2 macrophages surrounding the pancreatic tumor cells, indicating that it's the IRF7 modulations that is causing the cellular changes in the pancreatic tumor cells, at the same time causing high interactions to the pancreatic tumor cells also. So I hope you understood from the experimental results that it's the IRF modulations in the M1 macrophages that cause the cellular signalings or cellular changes in the pancreatic tumor cells. If you need to understand more about these experimental details and also the supplemental details that come up to these 4 genes, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.